Each of us is unique. We differ from one another in size, shape, face, and skin color. Beneath these surface differences lies a wealth of hidden variation that governs how we perceive the world through sight, sound, taste, and smell. Some of our most obvious differences are a reflection of our way of life, the food we eat, the environment in which we live. Some reflect our particular culture or upbringing, but many of our similarities and differences have their root in our biological inheritance. Each of us owes our existence to a commonplace but extraordinary event, the coming together of just two tiny human cells, one from each of our parents. The female egg, about the size of a pinhead, contains half the genetic information needed to create a human being. The male sperm contains the other half, crammed into an area many times smaller. Because of the complex way egg and sperm are formed, they are not simple genetic replicas of the parent cells. So when they unite at fertilization, it marks the start of a genetically unique life story. Now mitosis begins, a process of continual cell division from which our entire body is made. Deep in the nucleus of each cell lie tiny thread-like structures called chromosomes. They can be arranged in matching pairs, one partner from each of our parents. Every human cell contains 22 matching pairs, plus a pair of sex chromosomes called X and Y. If a mother's X chromosome is paired with a Y, the child will be a boy. If both parents contribute an X chromosome, the child is a girl. Sex is determined by whether a sperm carried an X or a Y chromosome into the egg. Every chromosome is a tightly wound package containing the genetic material, DNA. This molecule, deoxyribonucleic acid, specifies both our inherited similarities and differences within its simple but versatile chemical structure. The working units of information along the DNA molecule are the genes. These lie at the heart of our biological individuality and the inherited differences we see between people.
Some of the things we talked about is the ways in which we can see that we're different.
Alan Cadogan has been exploring these inherited differences with school children for many years.
Our eyes are different colours.
Our eyes are different colours.
Teeth have got flat. Our hair are different colours.
Our hair's different colours.
Our teeth are different colours.
No, there's another difference that we can easily see if we look at our fingers, because we can make fingerprints, can't we? Mm-hmm. We're all different as far as fingerprints are concerned.
You're different. I'm different. If you don't have if your fingerprints aren't the same, then you're not guilty.
Okay, so they can use our fingerprints to distinguish us.
Many of our most obvious physical characteristics have very complex modes of inheritance, but a few are inherited in a more clear-cut manner.
I want to ask you all at the same time do you see these little pieces of paper that I've got? I'm going to ask you each to put one little bit on your tongue, and I'll put a little bit on my tongue at the same time.
The ability to taste the chemical PTC, phenylthiocarbamide, is one of the simplest patterns of inheritance known. It's thought to be due to just a single gene found in two different forms or alleles. One allele allows the bitter taste to be detected, the other doesn't. Every person inherits one allele from each parent, and the particular combination received will determine whether someone is a taster or not. A person who inherits either one or two alleles for tasting will be able to detect the bitterness, while someone who inherits two alleles for non-tasting won't.
You can't taste anything. And some of you can.
There are different thresholds of sensitivity to the chemical, which may be governed by more than one gene.
This is like you're eating filter.
Now, be serious now, because some people can't taste this at all. Who couldn't taste anything? You couldn't taste anything. No. Could you taste anything, Cullen?
Yes, it tasted like some soil that was something horrible was put into
You do
it.
eat some funny things, don't you?
Yes, it was
Genes play a crucial role in all our other perceptions, but their effects on our colour vision are particularly striking. Inherited differences in colour sensitivity can give rise to what we commonly call colour blindness.
Okay, Craig. Now, here's...
It's being studied in various families by John Mullen at the University of Cambridge.
And what I'd like you to do is to find the colour amongst these that's most like this one and put it here. Okay? And then go back and find the one that's most like the one that you've just put down, so that you end up with a set of colours that change smoothly for you from one end of this little box to the other. There's no right or wrong about it.
9-year-old Craig Thomas has a particular form of red-green colour blindness, as his mother Elaine explains.
It was noticeable really when he first started school, and I went up to see his teacher to see how he was getting on at school, and she pointed out to me that she could see that he was having trouble with his colours and had wondered if he was colourblind, being as my father's colourblind. We often have trees with red leaves or orange leaves. We had a green monkey once after his visit to the zoo. And people's hair often comes out orange or green. And, you know, basically things are, the colours are very mixed up and unusual.
How's that?
That's very good. You're happy with that? There's nothing you'd want to change? Okay, well, I have one extra question for you. Where would you put that one? If you had to put it in, which one would it look most like?
I don't know which one. I think probably that one.
Okay. Right. Good. And if I said to you, which colours are warm and which are cold, would that make sense to you? Could you show me some that were warm and some that were cold?
The cold ones
Yes.
are that one,
Yes.
that one,
Yes.
that one, that one, that one, that one.
I see.
Those ones along there,
Yes.
and those three.
Okay. So which ones would be warm?
That one, 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 and that one.
Terrific. So you have actually arranged the colours in a very systematic way for you. You've got a set of warm ones in here and cold ones here and here, right? And this one you've put where it goes from cold to warm. Yeah? Good.
We can detect the three primary colours, red, blue and green, because of light-sensitive cells called cones packed in the retina of our eyes. The cones most sensitive to red and green light are coded for by two separate genes found close to each other on the X chromosome. This leads to a striking pattern of inheritance for red-green colour blindness.
Some 8% of men in our population exhibit some form of colour anomaly or deficiency, but the figure for women is much smaller, perhaps 0.4%. However, some 15% of women are carriers of colour blindness. That's to say, they're able to pass the gene on to their sons, even though they themselves don't overtly show any abnormality of colour vision.
To trace this pattern of inheritance in Craig's family, the colour vision test can be repeated for Craig's mother, Elaine, and for his grandfather, Gordon. The results for the 3 generations of the family can then be compared. Those of Craig's mother, Elaine, fall into a graded series of colours that change smoothly from one to the other. This arrangement is typical of a person with normal colour vision. But in Craig's case, colours that are noticeably different from each other are put very close together in the sequence. A similar result emerges for Craig's grandfather, suggesting that the basis for their colour blindness is the same. How could the trait have been passed on? Craig's grandfather Gordon is colourblind because one of the colour vision genes on his X chromosome is abnormal. Gordon passed this affected X chromosome onto his daughter Elaine, but its effects are masked by the normal X chromosome she inherited from her mother, who has perfect colour vision. The gene coding for normal colour vision is dominant to the gene for colour blindness. So Elaine appears to see colours in the normal way, but she is a carrier of the abnormal gene. Since Elaine's husband Martin has normal colour vision, what are the chances that any of their children might be colourblind? This will depend on whether the child is a boy or a girl. It turns out that any daughter will have normal colour vision. This is because she has a 50% chance of inheriting a normal X chromosome from both her father and her mother. Even when she inherits an affected X chromosome from her mother, her colour vision will still appear normal because of the functioning gene on her other X chromosome. The situation is very different for boys. Any son will inherit a Y chromosome from his father. This lacks any of the genes involved in normal colour vision. On average, there's a 50% chance that the boy will inherit a normal X chromosome from his mother and so have normal colour vision. But there's an equal chance that he will inherit Elaine's affected X chromosome. He will now be colourblind because there's no corresponding gene to compensate for it on his Y chromosome. This is just what happened in Craig's case. The colour blindness genes are just some of about 100,000 that are distributed among our chromosomes. In the complex process of meiosis, by which sperm and egg cells are formed, groups of genes from the parents are brought together in new combinations by an elaborate form of chromosomal dance. The chromosomes duplicate themselves and line up with each other in matching pairs, one partner from the mother and one from the father. A shuffling of genetic material takes place when the chromosome partners exchange pieces with one another, a process called crossing over. The chromosomes are then shared out in sets of 23, with each sperm or egg receiving just one partner from each pair. But it's a matter of chance which partner of the pair it will be. The likelihood that any sex cell will be an exact genetic replica of either parent is very small indeed. So each child that emerges from the coming together of a sperm and egg will be unique. We can see the visible results of this in a very simple, if unusual, demonstration set up by Alan Cadogan.
I'm going to ask you to stick your tongues out at me. You've got a pointy tongue, haven't you? You've got a pointy tongue as well, haven't you? I've been doing this sort of thing with young children for many years. But certainly at this age group, there's a tremendous interest in finding out the way in which they differ. Roll your tongues up like that. Can any of you roll your... Oh, that's smashing. That's lovely. Yes, you can. Apart from the usual things, like the difference in height and the eye colour and the hair colour, we want to get them to see which of them can do things like roll their tongues and folding their arms in different ways. Hold your right fist up in the air just so that I can make sure you all know which is your right. Keep your tight fist. Keep a closed fist, right? Keep it all the time. Now fold your arms like this. Make a big folded arm. Let's have a look at that. Now then, how many people, when they folded their arms, tucked their right fist down in? If you did, I want you to stand up. So if your right fist is in, stand up. Not many people realize that these things do have a genetic component. These things are inherited, like some of the chemical differences. Maybe things like the ability to taste certain substances or having a certain blood group. It's very interesting for them to look at some of these things as well as the more obvious ones. So that's a difference between us. Some of us tuck our right fists in, some of us tuck our left fists in when we fold our arms. Now, all of you sit back down in your places, back exactly as we were before.
The genetic basis for tongue rolling and arm folding isn't fully understood, but they can easily be scored on a yes-no basis. It takes just a few differences like this to divide a group into individuals with a unique combination of characteristics.
Just stand up, right? Stand up, please. Close together, nobody on the mats. And I want you to move so that all of the boys come onto this mat and all of the girls onto this mat. Now, please, do that. All of the girls are standing on the mat, all of the boys standing on the mat. Now I want the tongue rollers to come forward onto their mat.
The boys and girls can now be divided into two groups, those who can roll their tongues
Non-tongue rollers onto their mat.
and those who can't.
Stand still, Danielle. Now I want the people that fold their right arms in.
Alan then picks out children who fold their arms in a particular way, right over left.
Fold your arms, let's see your arms folded.
Finally, he can draw out those few individuals who, as well as the other traits, have a particular eye colour, blue.
Stand still, everybody, absolutely still.
The characteristics studied by Alan are relatively trivial, but the patterns of inheritance of certain traits have more critical consequences. Early attempts to transfer blood from one person to another often had disastrous results. We now know that the presence of distinct blood groups sets a limit on the number of people from whom any one person can receive blood. For instance, someone whose blood cells are group A can't give blood to someone of group B, because the recipient's immune system will attack the new blood and cause it to clump. Elaborate procedures are needed for testing and cross-matching the blood of both donor and recipient. The familiar ABO blood groups reflect biochemical differences on the surface of the red blood cells. A person's blood group depends not just on 2, but on 3 forms or alleles of a single gene. One form codes for substance A on the red blood cell surface, the other for substance B, and the third specifies neither and results in type O. We each inherit 2 copies of the gene for the ABO blood groups, one from each parent. A person with 2 copies of the gene for type A will be blood group A. One with 2 copies of the gene for type B will be blood group B. And one with 2 copies of the gene that codes for neither will be blood group O. We can predict the possible blood groups of children from the genetic makeup of their parents. For example, if a child inherits a gene for A from one parent and a gene for O from the other, its genetic makeup will be AO. But the effects of the O gene will be masked by the dominant A gene, and so the blood group of the child is A, just like its parent with a genetic makeup AA. Similarly, when one parent passes on the gene for B, while the other passes on the one for O, the genetic makeup of the child will be BO, but its blood group will be B, since B is dominant to O. But if the gene for A is paired with the gene for B, a different situation arises. Since neither A nor B is dominant to the other, the result is a new blood group, AB, in which both A and B are expressed. So we have 6 possible pairings of genetic instructions and 4 possible blood groups. The different blood groups can be used to exclude possible family relationships. For instance, if a child is blood group O, what genetic makeup would the parents need to have? The child must have received 1 copy of the gene for O from each parent. This could arise from a number of possible parental combinations, for instance, OO and AO, OO and BO, AO and BO, or 2 OO parents. At the same time, it automatically rules out parents that don't have at least 1 gene for O in their genetic makeup. For instance, any parent with the genetic constitution AA will only be able to contribute the gene for A to his or her child. Even when the contribution from the other parent is the gene for O, for instance in 3 possible cases, the child's blood group will always be A because A is dominant to O. A more direct way of establishing family relationships has been the unexpected spin-off from research by Alec Jeffreys at Leicester University. He has discovered patterns in the genetic material, the DNA, as characteristic of an individual as a fingerprint. He calls them genetic fingerprints.
They are patterns obtained on X-ray film. These patterns look rather like a barcode, say, on the library ticket. The pattern consists of a series of bands or stripes, each band or stripe being a genetic character. The pattern of stripes is extremely variable from one person to the next. The pattern is completely individual specific, with the sole exception of identical twins. They, and only they, have the same DNA fingerprint. Also, rather usefully and unlike ordinary fingerprints, these DNA fingerprints are inherited in a very simple fashion.
In each person's DNA fingerprint, about half of the bands come from the mother and half from the father. It's possible to produce identical fingerprints from a variety of tissues.
We need a sample of blood, or it can be other body tissues as well, but practically always we would use blood. That blood is then processed to extract the DNA, the genetic material. That DNA is then snipped up using enzymes which convert the DNA into millions of little bits and pieces of DNA. Some of those are highly variable from one person to the next. We then sort those bits of DNA out in a gel using an electric current to sort the bits out by size. And then most importantly, we then react that DNA with a radioactive probe which specifically binds to and highlights those bits of DNA that vary maximally from one person to the next. At the end of the experiment, we pick up that radioactivity using X-ray film, and it's a pattern on X-ray film, which is the genetic fingerprint.
So can the pattern be used to detect family relationships? This is the DNA fingerprint of a father, a mother, and their child. Mother and child have a number of bands in common. If we eliminate these common bands from the child's fingerprint, we are left with those that must have come from the true biological father. And these paternal bands do indeed correspond to the father's fingerprint. Here is the fingerprint of a man who was alleged to be the father. Comparing the child with the alleged father, they have a few bands in common, but not all. Only the true father has all the paternal bands in common with his child. What are the chances of a random identical match between two DNA fingerprints?
And that probability is absolutely vanishingly minute. In fact, it's about 1 part in a million. Or to put it another way round, if we were to analyze people at the rate of 1 million a second, we would not find a chance match like that until the death of the universe itself. It's that individual specific.
So our inherited differences account for many facets of our individuality, from our most obvious physical differences to the most subtle of chemical variations. Yet those aspects that most define us as human, our intelligence, creativity, social behavior, or culture, cannot fully be defined in genetic terms. But variety is the spice of life, and for our human diversity, it's a variety that owes much to our genes.